Hi, everyone. Thank you for joining us. We're just going to give it a few more minutes for more people to join, and then we'll go ahead and get started. As a reminder to everyone that since this is a focus group, we're really hoping that this is something that's very interactive. So we'll be monitoring both the chat and raised hands to see if there are any questions between the presentations that we have that are going to be ongoing. So feel free to do both. So with that, I am going to share my screen and we can go ahead and get started. Can you see this all right? Okay. All right, thank you again, everyone, for joining us today for the Knowledge Portal Focus Group. I'm going to go over some scientific updates, and then we will hand it over to our featured speakers. The first thing that I wanted to start out with is the NIDDK is having their 75th anniversary, and we are included in the anniversary collection, as you can see here. Thank you to everyone who made this possible. We're extremely excited to be involved in this, and you can take a look at the different articles that are involved. Ours here is the Accelerating Medicines Partnership in Type 2 Diabetes and Common Metabolic Diseases. And again, we're just thankful to be a part of this, and we're excited about the anniversary. Now I'm going to share some upcoming events that we're going to be attending. As always, we're going to be attending the American Diabetes Scientific Sessions in Chicago over in June. As part of this, we'll have our typical ancillary session, which we'll provide more details on at a later date. And we also have our ADA booth that we'll have, which will feature our various portals, including the CMDKP and a few others. One exciting thing to note is we'll have our talk for an oral abstract session, which will feature mechanisms linking adipose health with lipid and glucose homeostasis. And this is going to be introducing a brand new portal, so you're going to definitely want to check this one out. The portal will also be shown at the booth if you can't make the talk, but we're really excited to have this session as part of the ADA this year. Another conference that we attend every year that we'll be at is the American Society of Human Genetics. This is in October in Boston. We'll be showcasing our various portals, the CMDKP, the A2F, and various tissue portals. As part of this, we'll have both the ancillary session and booth, as I said, and we'll be hosting various community events throughout the week as well, so please be on the lookout for that. We're really excited as we'll be able to do so much more in Boston this year with everyone. Another event that I want to showcase is the Common Fund Data Ecosystem public webinar series. Our next session is going to be Friday, May 30th from 1 to 2 p.m. Eastern. And this will be focusing on sustainability and evaluation within the Common Fund Data Ecosystem. And presentations will be from the Integration and Coordination Center. And so with this, this session will be recorded if you're unable to attend as well. And more information will be provided on our site, which is cfdeknowledge.org. So we really look forward to that, and we hope to see you there as well. For upcoming meetings, we have today, which is the focus group session, and June 12th is going to be our next webinar. And then here we have the upcoming dates throughout 2025. So today we're really excited to be able to showcase how you'd be able to load your own GWAS and run LDSC or in a shareable protected environment. We're going to kick off the session with an introduction from Noel Burt, and then I'll hand it over to Drew Hite and Lucas Nguyen. Sorry if I pronounced that right. Please, like I said, feel free to ask any questions throughout the session. You can raise your hand, use the chat. We're just excited to have you all here today. So with that, I will hand it over to Noel.
All right. Oops. Sorry, we're not always in the same room, but can everyone hear me on the Zoom? See any faces or thumbs up? You guys can hear me? All right, beautiful. All right, so a lot of us are on the Broad today, which is great. I have all the presentations literally in the room with me, which always makes me happy and makes it much more of a cool experience. But sometimes we have hiccups in the audio. So I'm going to give I'm just going to set the playing field for us before I turn over to Drew and Lucas. As you know, I always like to do that to sort of contextualize why we're sharing you with what we are and at what time. And so I'm just going to give a little intro and then turn it over to the real exciting stuff, which you're here to listen to. So again, I always like to remind us that this group is across oceans and continents that build this resource. And I think that's incredibly important because that's how we build a resource that is hopefully serving the community in the best way possible. So, just contextualize what we started with a type 2 diabetes knowledge portal a long time ago, making summary statistics for GWAS data accessible to the world, right? So that was a simple ask and a simple need and a gap that we felt at the time. And that's expanded to common metabolic diseases beyond diabetes and also to the Association to Function Knowledge Portal, which spans common disease, right? And so we've done this by creating a community hub for any disease area community that's wanted it, but also integrating that with public and data from the community, a suite of methods and tools, and multifaceted access mechanism, whether it's through a nice visualization on a portal or through APIs or programmatic access or straight up old-fashioned download, right? So what does our resource enable? It enables something simple, which is I want to know what genes are involved in the risk of disease of a given trait that I study. Or I want to know, does the gene I study have a role in human disease? And that's easy to do, actually, if you're a geneticist. You can do it. It just takes a lot of work, right? You have to go to GWAS catalog, get all the data, download it, meta-analyze it, run these different methods, and you have to interrogate them for different kinds of evidence sources. And then maybe at the end, when you're tired, and you finally get to write a paper, you'll have a list of genes to investigate or waste of evidence for disease. But our job is to make that super easy for you by putting that all in one place and doing that with the best community data, the best methods, and the very specific and clear visualizations all run in a sustainable, scalable software environment. So I guess this sort of brings back to the pillars of how the portal's built. It's built based on community, data, and tools. And this is all about scientific stewardship, because most of what we represent is other people's work. But we also represent some novel applications, which is what you're going to hear about today, to enable others. So that's another way for stewardship, right? Stewardship is not just sharing results, but it's enabling things to become accessible to others so it can be used in other ways. So I'm just going to quickly review, and many of you know this who've been to our webinar, so I'm sorry if this is redundant, but I think it's very good to contextualize what you're about to hear, is that there are sort of three components of what makes our resource, right? One is the data, right? Genetic associations, functional genomic annotations, bioinformatic methods, and curated knowledge, things that are specifically curated by a community expert and brought into the portal. We bring these in through public ways, through consortia, through direct communication with wonderful investigators, and through our funded activities. Then same thing for annotations. So layering on annotations to those genetic data to sort of make predictions about what's going on at each of these variants or these genes. So we do this with Kyle Galton's team at the Common Metabolic Genome Atlas, CMDGA, where you bring all of the annotations that help you go from a causal variant to an organismal function, right? So all of those annotations really are really at the heart of metabolic disease. We have a lot of those in metabolic disease, but expanding it all across common disease as we go. But how do you put all this together, right? So these are bioinformatic methods, and this is why this is valuable. So we've encoded a host of bioinformatic methods, some of which are gold standards in the field, like MAGMA, for example, or VAP, right? But some of them are, novel approaches that we've developed here across the time that we built the portal as in response to things that are useful to you, like a bottom line integrative analysis for meta-analysis, right? You can submit your GWAS for any phenotype. If we have that phenotype already, we're going to produce a meta-analysis for you on the portal that accounts for sample overlap and does it in the same best practice standards of the field, right? So just to set that, this is all done in one infrastructure. And the idea being that we can build portals so people can visualize it in their data context, but also allow you to use these methods in a different way. So that's really the focus of today. So we have a lot of these methods. And if you work in the GWAS space, these are common knowledge, common terms to you, right? The ones in green are already in production, meaning that we run them on all of our summary level statistics and make them accessible in the portal. Ones that are in blue are in testing phase and we're starting to code and implement. And then we really want to hear your feedback about more. That's really how we've done this. But what's nice about this is that they're running every GWAS data set we get. But what we have not enabled is what if you just have that one investigator who's like, gosh, I have my own GWAS. I want to publish it, but I don't have the informatics infrastructure in my lab to run C2 CTS, to run COJO, to run stratified LDSC regression. I'd love some help with that. So those are some of the things that when you build a resource like that, you hear from your community, what their other limitations and gaps are. And so we think about stewardship for our portal, not only for the portal, but how do we can help communities do their research and make it easier. So to address other gaps, I just put these out there. These might be things that are useful to you. Like I have a GWAS, I need Magma Run, I need LDSC regression on just my data because I want to write a paper about it, but I don't want to worry about doing it. I also want you to do it in the best practices, or I want to work with you to tweak those parameters for certain methods so they're done a little bit differently. Mm-hmm. Or I have a GWAS all by itself, it's small. I want to QC it with my consortium members. I want to work together with them. And then I want to include it in their meta-analysis that they're doing all together. But we don't have a computational infrastructure to do that. We want to do that safely, even though some stats are easy to transfer, but we want to do it internally so we can make decisions and look through loci together. And then, oh man, we want to publish that whole unit of work externally in a UI along with our paper. Or I just want to share my GWAS statistics with the portal. This is the first use case, right? And I want it to be easy and I want it to be free. Well, we've enabled this one, but it used to be one of our computational biologists had to work with you, or we had one of our collaborators at GWAS catalog had to send us the data. Now we make it so you sitting in the comfort of your own home and load your own GWAS and run a suite of things. So that's what we're going to hear about today. And we always have to name things, and we always have to give it a catchy name. So we call this suite our GWAS or GWAS collaborative environment. And this is something that we're hoping to build out over the next couple of years, but we have some foundation here that you're going to hear about that I think is already useful to you as a community. The first is I got to load my own GWAS. So that's part of our KP and data registry. Drew's going to talk a little bit about that as we go forward. And then the idea is, why don't we start with one method? So we picked a pretty hard method. We picked a method that was already out there in the field, well-trained, well understood, kind of a gold standard for this particular activity, right? And we chose to harden it in software and run it across all of our data. So that's first thing. Then we wanted to say, well, if we did that, why don't we make it so you could run it on your own GWAS? So you're going to hear about that from Lucas, about how we've created a server for you to load your own sum stats, run them across LDSC regression. And then even better, though, to do this with a real vision, you want to make this a whole suite, a GWAS collaborative environment of other things, not only just one method. What about all of our methods? What about the fact that we have all these annotations for common diseases preloaded in this resource where you can take your GWAS and annotate them with? That's a value add. That's something that we would love to do for the community, even before you publish, even before you get things to the portal. We want to make it easier for you. We're thinking upstream of that. Well, what if you just have a GWAS before you've even done a meta-analysis? How do we help a community who may be smaller, who doesn't have, a huge, giant consortium or diagram, a really mature infrastructure to really do meta-analysis at high class, at scale, but knows how to do it? They just need a little bit of help. We've tried to address that gap too. So you're going to hear about those two things today. And ideally, we want to make this private at the beginning for people because they do want an environment where they, with their collaborators, can make decisions, view things together, QC their data before they make it accessible. And we found in every case that we've done this so far, everybody wants to make it accessible for many reasons. One, it's only useful if it's accessible. And two, journals make it required. So this is actually really an easy thing for people to ask. And private doesn't mean it prevents them from sharing. It makes them more willing to share, ironically. So with that, I'm going to turn it over to Drew, who's actually built a lot of the infrastructure you're going to hear about to talk about GWAS and its tools, and then you'll hear from Lucas. Stop sharing.
All right, let me share my screen.
Was I ever sharing my screen?
I think there was for a little bit and then it stopped.
Oh,
Yeah.
oh, lordy. Why didn't someone tell me?
You were ongoing and I didn't want to.
Okay, sorry guys. Hopefully I made it. Go ahead, Drew.
Yep.
I can share the slides after
Yeah.
if you're okay with that.
All right, so yeah, we have anyone in the chat, please let me know if you can't see my screen. So yeah, I have a browser window open to gwas-ce.kpnregistry.org. That site is publicly available. Anyone here can visit that themselves and should see the same view that I'm looking at currently. The first thing you may want to do when you get to that particular site is to upload a data set. So I'm going to click this button to upload. And you see that we collect a little bit of metadata. So we're going to specify a, uh, a data set name. So I'm just going to give it a simple name. Then we need to give the ancestry for our GWAS, and then specify a phenotype. And then you can either specify your in for the whole, either in your file or here as part of your metadata. I happen to roughly remember what the N is for this data set I'm going to upload. Then you specify your genome build, and finally choose your GWAS. So this tool will accept compressed and uncompressed GWAS files, tab delimited or CSV delimited. And once you browse to your local file, it will sample that file and identify what are the column headers. And then so the next step then is to map those column headers to the values that are required by um, uh, LD score regression, stratified LD score regression, I should say. So I'm going to quickly specify what those are. Let's see this. And once that happens, we will upload the file. Let's see. I think one more value is required. You can see sort of up at the top here that we've turned all the little buttons green. And at that point, we're ready to upload the data set. Backing up a second too, you can always click on actually, I don't see it here. It's on the other screen. I'll show you in a second. So go ahead and click upload. And this particular file is about 260 megabytes compressed, so not small, but not necessarily huge either. And luckily, we've got a pretty quick connection here, so it's going to finish up here in a second. All right, so now that that is uploaded, it immediately kicks off a SumStats process. So it's going to generate a SumStats file for that GWAS we just uploaded. Now that's going to take a few minutes, so I'm not going to hold everybody here staring at this screen. So what I'm going to do now is, this is kind of, as Noel said, sort of the easiest entry point for some of the things that we're building as far as trying to share our infrastructure, share our methods, share our data. But I'm going to show you now a more involved implementation of our data registry. This is a site that we've built for the Hermes consortium. And so in addition to, allowing them to access our infrastructure and our methods, we've also added a user management component. So one of the things that you may have noticed with the GWAS CE website is that it doesn't require a login, but you optionally can log in. So one of the implications of that is that everyone can see your analysis results if you choose to not log in. But if you're interested in using this tool, but only if you have sort of you know privacy protections, please reach out to us and we'd be happy to create a login for you. But anyhow, the Hermes wanted sort of two distinct types of users. They wanted sort of an uploader slash collaborator user and then a reviewer slash analyst user. So I'm going to log in as an uploader and show you what they're able to see. All right, so you can see uh, a six data sets that have been six GWAS files that have been uploaded, and they've all been uploaded by this particular user. So this user is only able to see, excuse me, their data sets. Uh, a few that they uploaded failed the QC process. A few did pass it. And so if it's ready for review, they can see the QC output. So Hermes has chosen a particular QC script that gets run as soon as the data is uploaded, and that will either pass or fail. And if it passes, the idea is that a reviewer is then able to um, review it and look at it in kind of more in a more granular fashion, and then give it a thumbs up or thumbs down. So I'll show you what that looks like really quick. So logging in as a reviewer right now. So now here you can see that there are data sets uploaded by multiple users in this case, not just the uploader. Um, if the reviewer wants to take a look at some of these data sets that have just recently been uploaded, they get a similar view to what we showed before, but they also have the option of rerunning the QC with different uh, parameters, and then also either passing or failing the data set. So they would look at the output of the QC and make sure it made sense, and either pass or fail it. And yeah, that's kind of a high level. Let's just check back in on our sum stats. I think it's still running, so I'll show you some more stuff in relation to what we've built for Hermes. So in addition to this functionality between this interaction between uploaders and reviewers, we've built meta-analysis functionality so that data sets that have passed QC are then eligible to be included in a meta-analysis. So we can click this button here, we can choose a trait, and then select data sets for that trait and include them in a meta-analysis. So we use behind the scenes for the meta-analysis, we use our Amazon Elastic Map Reduce infrastructure to do some variant scaling steps and eventually pass the results on to Metal and and run that in that Elastic Map Reduce environment, which sort of allows us to handle the data set sizes that we're talking about here when we're analyzing multiple gigabyte data sets, gigabyte plus. So then you can just specify a name for the analysis and kick it off. The meta-analysis step does usually take 25, 30 minutes, depending upon how many data sets you're including. So I'm not going to show that. But one other thing I will show is I'm going to log back in as the uploader user one more time. And I'll show you uh, the analogous step for uploading a GWAS in the Hermes portal environment. Uh, so here we recognize that it's Hermes is asking for quite a bit of metadata, as you can see. And so we thought it'd be nice to be able to reuse previously specified metadata values. So the first thing I'm going to do actually is choose the GWAS. And then the next thing I'm going to do is use a previous upload, and that will fill in most of the metadata most of the metadata with the exception of the dataset name. And so I'm just going to quickly fill in that name. All right. And oh, did it lose it? No, one more field I need to specify. All right, so that's going to chug along for a second. I'll just do a quick glance back. Okay, so some stats has now succeeded back in our original or excuse me, GWAS CE view. So the next thing you can do is then once your sum stats has succeeded, you're given the option of running stratified LD score regression. So we do that, and that's going to kick that off. Again, that probably takes another 3 to 5 minutes, so we won't wait for that, but we do have a data set that I've already shepherded through the whole process. In fact, it's the same one, so we're just going to click View Results on that. And so now you can see the results of the stratified LE score regression. So we have 5 columns, annotation, tissue, biosample, enrichment, p-value, and we're sorted by p-value from smallest to largest. And all these columns are filterable, so that you could, and actually, I will point out that the results make sense. We uploaded a GWAS for breast cancer, and the tissues that we're seeing are mammary gland, skin of body, lung, adipose tissue. So yeah, you can filter these results as well as download all the results to a TSV. And I think that's the gist of it. Any questions or anything I should follow up on?
I have a question, Drew. Are we looking for anyone else to test this? We have one group that's looking at this. We use it to load our GWAS data, ironically, but do we think we want to have some other folks who are interested in testing or something like that? Is it ready for that?
Yeah, it's definitely ready for testing, and we are. I think, as Noel mentioned, we're planning to add additional methods in the near term. We're going to be adding Magma by end of June. So yeah, we're definitely eager to get feedback and to sort of get affirmation, hopefully, that we're building something that's useful, and if not, get redirected towards where we need to go.
Beautiful. Other questions for Drew? And Drew, can we send out a link to this, or is that still something that we can? Okay.
Yeah, we can definitely
All right.
send out a link. I'll put one in the chat.
Yeah, we don't have any real help documentation yet for this yet. I must say it's very much off the press, but there's a little bit, right, Drew?
Yeah,
Yeah.
so
Okay.
what I was going to point out is that on this first page, the data set table page. So this is just showing the data sets that have been uploaded by the default user. But if you click on the help button there in the upper left, we kind of outline your steps. Upload, run some stats, which is actually going to happen for you automatically when you upload, and then click to run stratified LD regression. And then the last thing is, click on the results. We also have in this column over here on the right, the steps completed, sort of telling you where in the process you are. So hopefully it's pretty easy to use, but we'll wait to see what the community thinks.
Beautiful.
Can I have a
So
question,
what
please?
we're hoping is actually we get GWAS from many of you frequently enough that we might actually be asking some of you who submit GWAS to us who can help us with this. That's why Hermes actually was excellent.
There's a question.
Yep. Yeah, we can handle that. Does the LD score regression handle multi-ancestry? And how does that do that? Do you happen to know if it does that yet? I believe it does, right?
Yes, so you can
Yep.
specify mixed ancestry. Yep.
Yeah, you're right. So Kim, I think it is specific ancestries now that we have. Those are the ones that are in the knowledge portal as our consistent ones. So if there's other ones we need to add, that might be a case by case basis. But I imagine that would be straightforward to incorporate. That's a good question.
Yes, can I have a question, please?
All right, other questions before we go on to Lucas? Or we can also, if you have questions that are popping in the chat, we'll also go back to this because I think we'll have a little time at the end to talk about the entire suite of tools and how we make it more useful to others.
Noelle, I think we have
Thanks, Drew.
someone who would like to ask a question.
I'm
Go
gonna
ahead.
hand off to you, Lucas.
Hello, can you hear me?
We can hear you, but I don't think Noelle could hear you.
Okay, that's
Everyone can hear
great.
me now?
So
Oh,
I
Simon's trying
we
to ask a
have-
question. Hold on one second.
Yes, could you please ask the
Thanks,
question?
Kim.
And,
Who
then
is
we
trying to ask a question? Sorry.
can you hear me? I have a question about LDIC analysis. So I just if not if the LD matrix is required for this analysis. If this is a case, I guess I assume that you already have a
Can
pre-computed
you raise your hand if you're trying to ask a question?
LD matrix.
Oh, I can hear it. You
Yeah.
just need to turn on. One moment.
It's because I have their headphones on.
Can you repeat the last sentence?
the question was if the users would have to upload the LD matrix, and I think that they don't, right?
Yes, you
Yes,
don't
you-
have
We have
to upload your
pre-computed,
own,
yeah.
LD score panels. We pre-compute that for you. And I will show you the science just in a minute. All right.
Did that answer the
But-
question, or did you hear us?
Yes, great. Thank you so much.
Thank you.
Okay, so as everyone else introduced, I want to talk about GWAS and the science behind it, hopefully to entice you to use it more and see how convenient it is, and trust that we did our diligence when it comes to the science for the foundation of all of our tools. So Drew previously introduced you to the data registry, which is an infrastructure that allows user managed data. Each person can have their own login password where they store their own data in a safe and secure environment. And then GWAS is the data registry and bioinformatics methods that we want to incorporate together. And the very first line of product, as I already told, and Drew demo, is the stratified LD score regression method. But first, let's go into what is SLDSC. When it comes to genetics, the heritability of traits are affected by polygenicity and amongst other factors. But there is a particular focus on looking at the contribution of polygenicity versus the confounding variables such as population stratification. So Sullivan in 2015 developed the LD score regression method, and then later on developed stratified LD score regression, which extends the functionality of LDSC by partitioning heritability to functional annotations such as promoters, enhancers, binding sites, and then chromatin structures. It can even further up levels such as cell types or tissues. And this method basically answer the questions of what cell types, what tissues, or what functional annotations contribute the most to the heritability of a trait. And the reason why these methods are so established is that they utilize GWAS summary statistics instead of the individual genotype. And so they are scalable at large population size. However, running SLDSC by yourself requires complex environment dependencies and many data processing steps, including that LD score panel calculations and compute step, which is very like it turns out to be more complicated, and I learned that from experience as well. And that's why we developed the LD server. The LD server acts as a convenient way to do that. And our source of the annotations come from one of our most trusted collaborator, the Common Metabolic Disease Genome Atlas. We have an extensive set of annotations, and we compute all of the LD scores for those annotations before you even upload your data sets. So all you need to do is to bring your own summary statistics, and then you get from your own data sets to functional insight in just a few clicks, right? I hope that with this presentation, I can convince you that it's very accessible, efficient, and a very standardized method, and that we can convince you that we did our due diligence when it comes to the science and the method that we implemented on a web server. So now that that's the introductions of the web server, GWAS, I want to go into the validation of a method. We have previously introduced to you the bottom line GWAS meta-analysis that uses multiple large GWAS and then meta-analyze into one single summary statistics. And to validate the LDSC, SLDSC results from our GWAS web server, we want to compare that results to the SLDSC results from a bottom line meta-analysis. And this is a fair comparison because both of them will share the same set of annotations curated by our group from the Genome Atlas. So what are the logical steps here? Firstly, we want to show that the bottom line GWAS meta-analysis has the same discovery rate as any other large GWAS out there that are publicly available. And then we want to show that the SLDSC outputs for any random GWAS is biologically sensible. And then the very last step, we want to show that the SLDSC outputs are similar when using any random GWAS versus using the bottom line GWAS summary statistics. So let's look at the first step here, right? I have a plot here that shows when looking at the European ancestry and across over 200 traits, that the discovery rate for enrichment signals using SLDSC is similar between using the largest summary statistics available to us and then using the bottom line summary statistics. In fact, if you look at it, that the plot shows the discovery rate is more conservative when you're using the bottom line summary statistics compared to like regular large summary statistics. And then the second plot here, for most traits, the rank correlation of significant tissues as shown by the Spearman's correlation with coefficient larger than 0.9 shows that the top enriched tissues from SLDSC results are similar when using the largest summary statistics versus the bottom line summary statistics. So I hope that can convince you that comparison between any random summary statistics, the bottom line summary statistics makes sense because they correlate well together. And now we look at some example SLDSC results at the individual summary statistics level and compare that to the bottom line summary statistics. I have here, if you look at the top left, a summary statistics that came from the Veteran Affair Million Veteran Program. This is for the European ancestry with type 2 diabetes as the trait and around 450,000 population size. And this is like the summary statistic that we have uploaded straight to the GWAS server, and then the results that will be displayed. And when we translate that to the table and compare it to the bottom line results, you can see that the enrichment coefficient and the p-value are very similar, and that the tissues and the cell types / biosamples are biologically sensible when it comes to type 2 diabetes, which is a very well-known and well-studied trait. Another example that we want to show here is a summary statistics, also from the European ancestry with the heart failure as the trait, also extremely large sample size. This is the results that you will get from the GWAS web server, and translating to a table and comparing it to the bottom line results analysis. Once again, you can see that the enrichment coefficient and p-value are very similar. And previously, we have introduced you to the Association to Function Knowledge Portal Network. And now we want to show you that the method that we have implemented on the GWAS server is the same as the method that we have implemented on the A2F portal. And so now we'll be using our actually bottom line summary statistics, uploading it to the web server, and then comparing it to the results display on the ATF portal. Here is the bottom line summary statistics for the European ancestry, heart failure, and this is the results that you will get from the web server. And when we compare it to the portal results, you can see that it's very similar, almost exactly the same. The minor difference here is just the technicality of the implementations on different infrastructure, but the discovery and the tissues and biosamples and the insights are exactly the same. And so I hope with that validations and all of the ease of use through the demo can convince you that you can safely upload your summary statistics to our web server, GWAS, and then get functional insight in such a fast and convenient way. And in the future, like Noel and Drew have discussed, we are planning to add more bioinformatics tools, established ones like Magma, other methods of LDSC, like cross-strait LDSC, and then Kojo, and hopefully we will see you coming back to use our web server. Thank you.
Excellent. I'm going to take my computer off mute so that I can hear everybody.
Yeah.
It's all right. All right. Can folks hear me still? Questions? Feedback? What method should we do next? We got Magma in the pipe. What other methods would be valuable to the community? Someone have a comment? No? Well, I see Ryan Welch on, oh, can you see the plan list again? Yes, I think you can. Let me show my slides. Haha, finally. No, it's funny. Oh, man. You guys are so used to me talking so fast, you probably don't even know what I'm saying in my slides. Here are the slides. So the methods are here. Here we go. All right. So right now the server only does LD score regression. So all the other ones that are in green are already implemented in the aggregator. So those are easy. Those are, well, easier, I should say. Sorry, Drew, to encode and run on in GWAS. Then the ones that are down below, I think we're trying to do in parallel, like on the portal and through GWAS, such that you can do both mechanisms. I think working with our Michigan colleagues, I see Ryan's on the call. You know, we also already have a visualization for our co-localization through CoLocus. I think that's another direction we're hoping with co-localization methods, and then Suzy, Kojo, whatnot, making and other fine mapping methods, integrating those so that we can actually then visualize them with CoLocus and then allow others to visualize them. So I think that's something we're hoping to work on with our Michigan colleagues. Let me look at the chat. Sorry, I can't do two things at once.
I have one that is on here from a direct message. Can you share the strength of LDSC method over bottom line GWAS method?
Yes.
Yeah, we can do that. Activity with contact, good one. Yeah, actually, Kim, that's one that we had on the site and we have not. Circle back. I like that one. ABC. That's a good one. Add it to the list. Lucas, you want to answer that where the LD is calculated from?
Apologies.
Yeah.
Yeah,
Yep. Hold on. Go.
Okay. So we calculated the LD score from using the original method published in 2015 with the Finnegan lab, using an external genotyping panel from the 1000 Genome Project, specifically to each of the ancestries that you see available on the web server right now. So each of them have a set of files, stands for the LD score for each of the ancestries, and then we use them to calculate the enrichment values whenever you upload summary statistics.
Will there be an option to use a non-1000 genomes KG reference of our own? Good question.
I mean, I think it would be possible, but it would definitely be faster, and it is already recommended since 2015 and 2018, the later methods that recomputing an LD panel is not necessary, but I guess if it's possible, we can
Yep.
implement such an option.
Yeah, it's not a bad idea actually to have as a backup,
Mhm.
particularly for, you know, certain consortium will want to do it a certain way. Just think what you're alluding to. Oops, I can't be heard. There you go. Come back.
See,
I see.
You guys can- you want to unmute yourself, sorry. We're doing a lot of mute dancing here. Yeah, that's a good idea. And I agree, Kim, that might be useful for us to have as a backup plan for people, given certain consortium may want to do it a certain way. Other comments, thoughts, suggestions? We're hoping by ASHG to have Magma in to also, there's another community that luckily we're actually really fortunate to be working with, the Skin Genetics Consortium. You know, it's a smaller group of consortia members, and they really were keen to see like, oh, wow, we need a place to do all the things that you just said. So we're going to be working with them. You'll hear about them, hopefully how that's going with them in the fall as well. So, and we're anxious to do this with you all if it makes it easier for you, or if you think, you know, that we should change our direction in terms of where we're starting. But I like the idea of ABC as a value add to this. You're going to soon see this actually part of the knowledge portals. Like when you go to the knowledge portal, you can actually get a drop down menu, select data registry, register your data. We're getting there. But right now we want some early adopters to make sure all the kinks are worked out. Luckily, the Hermes group from Heart Failure has been very gracious in testing it all with us and finding the goods and the bads and the really helpful pieces. Any other questions or comments? All right. Well, Drew, Lucas, that was excellent. Thank you so much. Sorry, I didn't share my slides, everybody. I sent a link. You could have them there if they're useful. It's basically a lot of what Drew and Lucas went over, but just gives you some high-level summaries of it if it's useful. When is our next date, Mackenzie? Did you already go over that?
I did, but
Okay,
it's in
great.
June, so it's going to, can everyone hear me okay?
They can probably hear it's fine.
Well, and we'll have a webinar, so we look forward to having you all there, for that, and we'll send more details as we get closer to.
And also, people have any ideas for webinars? We're always looking for new ideas or things that you would like to hear about, that we haven't covered recently. So please just send us a note or email me or Mackenzie. Hopefully you know all our email addresses, or just our help at cfd.org. Not CFD, KPN, too many websites, but you know what I'm talking about. All right. So thanks everybody. And we'll see you in a month, and everything will be posted for you to visualize and access offline if you so choose. All right. Thanks to our speakers.
Thank
Take
you,
care, everyone.
everyone.
Bye-bye.